Hi, my name is Kaiwen Zhang, and my presentation today is about a new approach that we recently developed to acoustically measure the local stiffness within embryonic tissue. From an engineering perspective, one way of defining stiffness is to use elastic modulus, which characterizes a material's resistance to deformation under load. The reason why we focus on stiffness is because it's relevant to some major fields in research. One is morphogenesis in developmental biology, also what Raymond had just talked about, where it tracks how cells initially divide, migrate, and differentiate to shape organs in the embryonic tissue. In literature, although stiffness has been shown to influence morphogenesis, we don't know the mechanisms involved. What we do know is that biological tissue is anisotropic, meaning that at different locations within the tissue, the stiffness is likely to be different. We had previously investigated stiffness within the mandibular arch of the mouse embryo, which is around the lower jaw area, from when it was a cylindrical shape till later acquiring a sort of a teardrop morphology. And we found the values to be different along different axes. If you look at the boxed areas, as the arch was undergoing changes, the middle portion sort of narrows in the anterior-posterior direction, but along the proximal distal direction, the tissue elongated. So this is a potential implication that local stiffness can regulate organ shaping, raising the possibility to catch and treat developmental abnormalities in the embryonic stage. The other use is to help identify tumors in cancer research. Cells tend to migrate towards stiffer areas, and increased cell tissue stiffness is sometimes an indication of solid tumors because healthy tissue is often in smooth layers, but cancer cells tend to modulate surrounding cells to form lumps. Clinically, by creating stiffness plots in addition to existing examining methods, there is potential for physicians to locate and isolate areas of interest to proceed with targeted treatment plans. So historically, the governing steps to measure stiffness are firstly actuation and then visualization. Actuation is that some sort of external force is needed to introduce a local displacement, which is then subsequently recorded to form images. So following these steps, previous work has been concentrated on plane or surface measurement, which gives low throughput because it is performed on single or only a couple cells at a time, but never on multiple cells in a bulk tissue. For example, atomic force microscopy is a tool to map stiffness across the surface by indentation, but it takes measurement point by point. Micropipette aspiration is often applied on a single cell by creating a suction. Magnetic tweezers use magnetic beads that needs to be deposited into the sample prior to actuation, which is invasive because it creates little punctures to the sample. In addition, each bead is limited to one specific location, which also gives you a low measuring throughput because if you need to measure multiple locations, you would have to deposit multiple beads. The emergence of MRE that uses MRI in conjunction with acoustic waves is highly prominent, but it suffers from the problem of high cost that is not suitable for a laboratory setting. So what we're hoping to achieve is sort of simultaneous point measurements instead of point-to-point in a non-invasive manner at low cost. If the cells within a tissue sample can be examined simultaneously, we would be able to increase the throughput. And we found that a combination of acoustic waves, which generate acoustic radiation force as the actuation method and light chain microscopy as the visualization method, has the potential to overcome the limitations we're concerned about. We then designed an experiment of a couple of parts. Start with the middle portion here. A mouse embryo will firstly be stained so that later on the nuclei of the cells can be distinguished because they would present a certain color. We prepare the embryo in agarose gel in a glass capillary that is fixed within the workspace of the microscope. We've chosen disc piezoelectric to deliver the waves. So how a piezoelectric crystal works is that when it's actuated, the thickness changes because it elongates and contracts repeatedly along that direction and generates continuous waves that travel away from the crystal. A piezoelectric can fit within the capillary because of its size and shape, and the frequency of oscillation is also adjustable depending on the experimental need. But to signal the piezoelectric, it requires a function generator that is able to output adjustable frequencies. We've chosen one that can generate a maximum frequency of 10 megahertz, which is a pretty safe maximum, and also sine waves, which are compatible with the piezoelectric. We also need a voltage amplifier, which controls the intensity of the waves coming out of the function generator so that it is large enough to power the piezoelectric. Once the sample is under acoustic influence, the light stream microscopy illuminates the cross-section of the sample such that the direction of local displacement lies on the illuminated plane. We can then visualize the previously stained nuclei of the cells within the sample. There are two parameters that are important in defining the wave because we want to simultaneously take measurements of multiple cells. We should minimize the difference at various locations to minimize sampling variance. For waves propagating, the speed is only relevant to the intrinsic material property. So the speed v here is fixed because the tissue properties do not change over the course of an experiment. So then the relationship here suggests that the higher the input frequency, the shorter the wavelength, meaning that the cycles are closer together and the waves propagate through more nuclei, minimizing the variance. However, higher frequency increases the loss in wave intensity, which determines how powerful the waves are. More intense waves have higher amplitude and can exert larger pressure, so the change in displacement is larger. We did some simulation in COMSOL multiphysics. We just wanted to have an idea of how the waves propagate, but it also enables us to adjust the parameters virtually to simulate the potential effects. A challenge that we currently have is the attenuation in wave intensity. A concern is that cells may be receiving very different external loads because of the attenuation. But instead of attempting to quantify the attenuation, we more so want to know if the percentage attenuated is within an acceptable range. So maybe we can neglect it for now. What we came up with is a test with 1% agarose solid that mimics a homogeneous and isotropic tissue sample. We'll place beads close to and far from the piezoelectric and record the local displacement of each bead. If we notice that the changes in displacement are very different, attenuation then has to be considered. Then we'll backtrack to calculate and correct the percentage attenuation, assuming that the decay is linear, because as I mentioned, the material the wave attenuated in is isotropic. So moving forward, we're able to create a 2D stiffness map using local values. If we put on a color scale, the plot becomes what is called an elastography that some research groups are actually currently working on. Furthermore, the illumination can happen at a different cross-section of the embryo, so we can illuminate multiple cross-sections of the sample to form a volumetric stiffness map. Additionally, if we take timescale into consideration, embryos and tumors can be imaged at different stages of development. This video here shows the progression of development of a fly embryo over time. So on top of that, perhaps a progression of stiffness can be added to further facilitate our understanding of its relationship to either its embryogenesis or progression of diseases. Lastly, I would like to thank my supervisor, Min, my PI, Professor Sun, Dr. Hopian, the MIE undergraduate office for the guidance and funding, and UNER for organizing the event for the opportunity to share my work. I'll be happy to take any questions.
Thank you very much, Kaiwen. So if we have questions from the judges, can you please just unmute your mic and ask away?
Hi, Kaiwen. Great presentation. So I really liked the assessment of stiffness within the human embryonic tissue. I was wondering what other tissues would this be important to measure, and can we apply atomic force microscopy to other more complex systems and complex tissue systems?
So what it offers is that, first of all, it's non-invasive, so it would be suitable for human. Actually, it's just it's taken from the idea of ultrasound, which uses acoustic wave to probe into organs. But the problem with ultrasound is that it offers a very poor resolution. So we're trying to solve that with a light sheet microscopy that can offer a very fine resolution down to a cell level. So a combination of a good resolution and non-invasiveness, I guess you could you could apply to whichever tissue of your choice.
Great, thank you so much, Kaiwen.
Problem.